Hello everyone and welcome to a brand new video on rhodopsin based photoreception. So we're going to talk about how rods and cones work. So in this video we are going to discuss the anatomy of rods and cones. We're going to look at the structure of the rhodopsin molecule. We'll look at dark current and we're going to look at how that's generated. And then we're going to look at the steps in actually rhodopsin based visual physiology. So we like to outline things and always like to say this is how we started off, this is how we're going to make it work. So let's go into it and discuss it. And as we said, the first thing we really want to talk about is the differences in rods versus cones. So let's talk about over here, we'll draw out the anatomy of a rod, of rod cells. So we'll look at rods and we'll look at cones. And when we look at rods and cones, what I really want to make sure we understand is the physiology will be the same for both. They have slightly different structure and function a little bit differently, but they use the same basic physiology to function. So once we get one, we'll use rods as our example because it's a bit more simple to explain. So let's draw out a rod, and this will be representing my normal rod cell. And we'll sketch this. And then over here, we'll sketch out a cone cell. And in the cones, all right. And then in cones, let's draw that. All right. So what we've done is drawn a basic rod and cone cell. So with this, let's go over the basic structure. Now, what we want to do, kind of start off, is talk about two basic segments that these have, two kind of base segments. First, there is what is called an outer segment that these things have, an outer segment, and an inner segment. Now, it's important to note that the outer segment is up against the pigmented epithelium and the inner segment is facing towards the bipolar cells. Okay, so let's talk about some other structures. Let's talk about these things right here. These are the discs, the visual discs. So what about discs? We want to talk about the stalks that hold them on. We want to talk about the cell bodies. I like to just mention those real quick. And then lastly, the synaptic terminals. Let me erase that and start over. There we go. I'm not going to just restart the videos because I did that. Okay, synaptic terminals. All right. So these are our basic structures of rods and cones. Now what we want to do is if we are going to, basically what we're going to do here is imagine that I've got a rod cell and we had talked about the discs. So what we're going to do is we're going to zoom in and talk about these discs and get an idea what's going on in one of those. So let's say that I've got a disc and I were to zoom in on said disc. What are we going to see in here? Okay, what is it that we're going to see if we zoom in on one of these discs? So here I have part of the disc. And then what I'm going to see is this globular protein embedded in it in these discs. Okay, so this is representing the disc structure here. Now let's imagine those little that little blue glob. Let's imagine there's many of these studded on these discs. Okay, we just kind of imagine there's many of them around. Now, inside this thing, we're going to talk about what it is in a minute. There will be a molecule, and I'm going to give it its basic shape here. The basic structure that this thing has is going to be something like this. So what are we dealing with here? Well, what we have, first off, is this blue structure. This is a protein referred to as opsin. And what opsin does is it contains a pigment molecule here called retinal. Combined, when you take opsin and retinol and combine them together, you make a structure, a thing called rhodopsin. So what rhodopsin is opsin and retinol. Retinol is the pigment molecule contained within this protein opsin, and this is going to be used in the visual transduction. Different opsins are going to absorb different wavelengths and let different wavelengths in to activate this retinol to activate a series of events. So this is our rhodopsin molecule. Okay, and this is basically what's going on, is they inhabit the discs and scattered all throughout. Okay, now so imagine them stuck in the membrane on these discs. Okay, so what we want to do now is go in and understand what is happening in the dark. It's something called dark current. So let's talk about the dark current. What is it? Okay, so basically with dark current, this is when you are in the dark, okay? You are in the dark. So we're going to talk about when we're in the dark, what's going to happen in the dark. So starting with this, what we need to do is we're going to need to draw a rod cell. We're going to draw them just a little bit larger here because we're going to need to have some physiology going on inside of it. And I'm going to leave a little gap right there. Let me draw all these discs so we kind of have an idea a little bit about that going down. All right. And then what we're going to do is we're just going to draw a little bit of a bipolar cell here. I've been trying to keep the text consistent. Okay, bipolar cell. There we go. Okay, so we got a bipolar cell here. So what we're going to do is in this rod and cone, we have all these discs inside this rod cell. We're going to use rod as our example. Now, let's say we're in the dark. It means there's no light coming in to stimulate this. So what's going to happen is we are going to have these proteins that embed the cell membrane. There are many of them, many of these protein gates, channels. And what they're going to do is they're going to be able to open and close based on whether or not this cell has absorbed photons. So they will be open in the dark. Now, how is that? Let's imagine this is like a door, and we have a door stop, okay? And this door stop that's going to be on it is going to be important. We're going to name it first. This door stop on here is called CGMP. So what these proteins are here are CGMP gated. They are CGMP gated ion channels. Now they're going to we're just going to basically call them CGMP gated sodium channels. So what's going to happen here is that because all cells, all excitable cells, all cells have this protein in them, who we mentioned many times on my channel, is going to bring in 2 potassium cations in and kick 3 sodium cations out, sodium potassium ATPase pump, you always will have a very high sodium concentration outside of cells. So because of this, what the sodium ions will do is they will flow in this through what's called the outer segment out to the inner segment. And so sodium cations will flow in the outer segment and out the inner segment. And what this is basically going to do here is it's going to make the inside of this photoreceptor quite a bit more positive, depolarizing it. Now, as it depolarizes it, if we were to basically kind of understand what's happening here, is that over time, and again, over time, we will see a voltage change in millivolts in the level of this ion. And what's going to happen is it might start here and then rise to a maximum level. It typically is going to be down here at about -70 when it's working, but when we are suppressing it, it's going to stay about -40. So what we're going to be doing is this is working at -40. So this is going in a more positive direction. -40 is more positive than -70. This is dark current. This is going to lead to a depolarization. Now, with depolarized cells, one of the things they tend to do is we know that a cell in its synaptic terminal will hold vesicles. And we know these vesicles contain in them neurotransmitters. And these neurotransmitters, for example, will be released through exocytosis. Now, in this case, the neurotransmitter in question here is called glutamate. And this glutamate is going to take and inhibit this bipolar cell when it's receiving it, and this is what is called dark current. Is in the dark, this is doing this. Now, there's two good reasons to do this. Number one, you are actually asleep less than you're awake, so you save glutamate. But also, if a photoreceptor worked like neurons did, instead of making an action potential, they make receptor potentials. They work when you need them to work. This also means you won't be blind during the absolute refractory period of an excitable cell. So this is what's happening here. So what happens in the light? What happens when I turn the light on? Now, what we need again is our photoreceptor. And we're going to have our rod cell as our stand-in for photoreceptors. And we're going to leave that a little bit open. Now, I always add all these discs. I know they can get in the way of the drawing, but I really want those guys there to help understand what's going down. Now, we talked about dark current was happening. So what we said was going on, remember, is that you've got these cGMP gated ion channels letting sodium, for example, in. And that was what was going on. And we said that basically you have this molecule here called, sorry, I forgot to change color, called cGMP. And what we said that cGMP is like a doorstop. Okay, it's going to be like a doorstop. Now, this doorstop constantly allowed the sodium ions to enter the outer segment and flow in out the inner segment, depolarizing that cell, and that was called dark current. Now we want to stop this. So what's going to happen is, let's say here the light happens, and we have some light coming in. The light comes on. And that light strikes one of these, starts to strike the photoreceptor. Light comes in. What's going to happen? Well, let's zoom back in on our little discs here and talk about what is happening inside of that when we see. Let's look at the cellular physiological change that happens here. And I'm going to make that quite a bit longer here. And what's going to happen? So we have our pals here that we just learned about, our friends that we just learned about. Remember, we have opsin and we have retinal. And together, this is called rhodopsin. Now, what I want to help you understand about retinal is retinal has two conformations it can take. So what's going to happen is right now, retinal is in what it's referred to as its more crooked state. So if I were to draw him here, we're going to say he's in a less linear crooked state in what is called the 11 cis conformation. It's 11 cis shape. Now, what I want to help you kind of understand, let's imagine that you have somebody, your sister, maybe is a little bit crooked, maybe a little bit untrustworthy at a certain point in their life. So they're a little crooked. But then they see the light of about the error of their ways and they straighten themselves out. So what can happen is it goes to an 11 trans, which is a more linear conformation. Now, how does that happen? How do you go from 11 cis to 11 trans retinal? Well, that's going to be light helping us do this, okay? So our 11 cis versus 11 trans retinal, light's going to do that. So what's going to happen is light's going to come in and it's going to strike this. And what's going to happen is this light is basically going to give the energy needed to be able to change the shape of this molecule. Light is going to be able to affect the needed energy, and it's going to linearize this. Now, let's just say, let's use an analogy I like to use in class. When I'm teaching this, what I always say is, let's say you guys, it's on a beautiful afternoon and you're studying for your anatomy and physiology exam. And so you go take the door, because it's beautiful, and you prop the door open. And what you do is you go to study and you're sitting at the desk studying and the breeze is coming in, but the breeze is coming in so much it's blowing your notes and making it impossible to see the notes. So you stick your finger out at somebody, you straighten your finger in your arm, you stick your finger and point to somebody in the house and you go shut the door. Well, that's what happens. That's going from 11 cis, 11 trans. You point to somebody and say, you go shut the door. Well, the person you asked to go shut the door, let's say you ask this individual, go shut the door, and this person's name is transducin. Transducin. What's going to happen here is that when we go from 11 cis, 11 trans retinal, this releases enzymes that basically activate transducin, okay? It activates transducin. Now, what transducin is going to be able to do, it's going to be able to activate an enzyme that we have inside of our cells, an enzyme here called, I'm going to write PDE. Let me just give myself a little bit more room. One of the reasons why I use the smaller pen is it's a little bit easier to see. PDE, phosphodiesterase. Now, what's going to happen is transducin is going to activate PDE. And this PDE, basically what it's going to do, it's an enzyme, phosphodiesterase. And when it gets activated, he comes up as an enzyme, PDE, phosphodiesterase. So what's he going to do is he's going to consume and start to break down our cyclic guanosine monophosphate, our cGMP. Now, it so happens cGMP is right here. Right here we have a cGMP. So what we're going to do is we're going to gobble it up. That means we're going to break down that cGMP. And when we get rid of it, the doorstop is being kicked out and this gate's going to close. And when this gate closes, the sodium can't flow in anymore. If it can't flow in anymore, okay, it can't flow in anymore, this dark current is going to diminish. Now what's going to happen is, we talked about our good friend bipolar cell was here receiving glutamate and being inhibited. Well, this glutamate stops, and what's going to happen is this cell here no longer releases glutamate. Why? Because the sodium potassium ATPase is pumping 2 potassiums in and 3 sodiums out. It's losing a lot more cations than it's gaining. And what's going to happen if we were to graph this, let's draw that graph here, is our voltage in millivolts over time is now going to see a plummet. It was up here about -40. And it's going to drop down to -70, and we're going to hyperpolarize it. And when we hyperpolarize it, you no longer release that glutamate. And since that glutamate's no longer being released, we now remove the inhibitory action, and bipolar cell can fire and let the ganglion cells know we've seen. And that's because cGMP went and broke down, it was broken down by PDE. PDE acts as an enzyme here. PDE, phosphodiesterase, breaks down that cGMP, and that's going to close the gates, okay? And this is the basic process behind rhodopsin-based visual transduction and how it works. So basically the deal is at night you have the dark current flowing, which is going to release glutamate, inhibiting bipolar cells, inhibiting the optic pathway, whereas then we're going to come in and we're going to have a light, photons from light are going to start to come in, hit these discs, and they're going to activate retinal to go to its more linear, from 11 cis to a more linear 11 trans, which activates transducin to activate PDE to then break down cGMP, and that allows us to see. And the analogy I always say is, you're trying to study on a beautiful day, you've propped the door open to let the breeze in, but the breeze is blowing too strong, so you reach your arm out and point to someone, let's say in your house, and you say, you go close the door. That was transducin. You ask them to close the door. Transducin doesn't want to do it, so they ask someone else in the house, you go close the door. They walk over and kick the doorstop out, the door shuts, and you're allowed to stop letting the flow of sodium coming in. The breeze is no longer blowing in. You can see your nose. I hope this helps to understand rhodopsin-based visual physiology in a way that can be clear and understood. There are more details, of course, to discuss. If you're looking for a discussion on things like guanylyl cyclase and how that plays a role and some of the other extra things, let me know, and I can do an extra in-depth video on this where I go more in detail. But I think this is a great detail that really would be good towards the more detailed leaning anatomy and physiology instructors, or even human phys, and some good foundational concept for this idea, this concept here. I am going to also have a video that goes with this about, so I urge you to watch it as well, on the histology of the retina. But guys, thank you so much for watching, and as always, don't forget to like, subscribe, leave a comment. Let me know what you would like to see in future videos, but it's just as always, thank you so much for watching. I'll see you in the next one.